Yes, I mean, I'm really looking forward to kind of this conversation because it's on a topic I've been interested in kind of as an outsider for a while. And I think from what I can tell, you do pretty unique research in this area by adding kind of a very different perspective on topics that I've heard about, done in other species, that kind of stuff. So yeah, I'm really looking forward to kind of exploring the topics, and especially what I'm interested in talking about more is kind of how you started working with bats and talking about bats themselves, the ecology, what bats do, how they live, and that kind of stuff. And I thought maybe the easiest way to kind of do that is to kind of track your education until you started working with bats, because from what I can tell, you didn't start off that way. So maybe as a kind of first question, I saw you studied physics for your bachelor's. So kind of why did you study physics? What do you think you were going to do with that?
So as a kid, I really loved math, loved physics. I also loved zoology. I was watching BBC documentaries, David Attenborough and such. So my mom always thought when I was a kid that I'm going to go study biology, zoology, something like that. And I surprised her by going to physics. That's because, I love that topic as well, especially math. So, of course, the funny thing is that after studying physics, then going to neuroscience, and ended up doing research on neuroscience in bats, including in natural behavior. So ended up coming back to zoology in a way. So I guess you can't escape your childhood loves.
Yeah, but you wanted to, did you have like a particular career in mind when you started or during your degree, or was it just you were interested in the topic and let's see what happens?
No, I loved physics. So I studied physics, and then I went into the army, which was in Israel, everybody have to go to the army. So it naturally introduces a gap in your academic career, because I did my PhD, my physics undergrad before the army. So then I went to the army, and then this created a gap. So it gave me an opportunity to consider what I want to do next. And then while I was still in the army, I was more than 5 years in the army, I was exploring all sorts of directions. I decided I want to go into biology, that my quantitative background is useful for biology. Today, it's very common to think this way, but back then it was a bit less common. So for a year, I took all sorts of undergrad courses in, cell biology, genetics, biochemistry, stuff like that. Then for another year, I took courses in neuroscience. So for about 3 years, I was meandering until I decided that neuroscience indeed is what I want to do. So it was a bit of a meandering to even go into neuroscience. And then I did a PhD in neuroscience, but not in bats. I actually worked on auditory cortex in cats. Today, almost nobody studies cats, but back then, in the 90s still, and of course before that, cats were the standard animal model for sensory neuroscience, and all the big discoveries of Hubel and Weizman in visual neuroscience, and then likewise in the auditory domain. Everything was done in the 50s and 60s and 70s and 80s in cats, almost everything. I mean, I still remember as a PhD student in the late 90s, hearing for the first time about a lab that started to study auditory cortex in mice. And I said to myself, this is weird. Why mice? They barely hear. This is a weird animal model to use. Nowadays, everybody's doing everything in mice, but back then it sounded like a weird idea. And so, I completed a very successful PhD, successful in terms of findings and publications, and could have easily continued, working in mainstream neuroscience, let's call it this way. But I decided to change direction, for my postdoc, and for a number of reasons. So one is that during my PhD, I found some effects in the auditory cortex of cats that were sort of memory related or were dependent on the history of the stimulus. So I got interested in memory, started reading about memory, and then of course you bump into the hippocampus and you read about place cells and all of that. Grid cells did not exist back then. And I thought to myself, wow, this is really amazing. Of course, I heard about place cells in courses, etc., but it's different when you start reading papers, actually. But then I saw there's so many labs working on place cells in rodents, and I guess I just want to do something else. If everybody are working in rodents, I just want to do something else. That was sort of one direction of how I started to look for which would be an interesting species to work on. Then I also, because I worked on auditory neuroscience, I knew about bats. I didn't work on bats, but bats are actually quite a substantial or an important animal model in auditory neuroscience. So I was, seeing lectures, posters on bats, so I knew about them, more than the average neuroscientist, let's put it this way. And then the third thing, I took a course in neuroethology, and this really blew my mind. I really decided that looking at the neural basis of natural behaviors, that's the way to go. And so, it was a convergence through several directions, and it took me several years. It wasn't like an overnight decision. It took me a couple of years to converge on this. And, there's also, if you start thinking, if I want to study the hippocampus and want to study the neural basis of behaviors, but I don't want to do it in rodents because everybody are doing it in rodents, which would be a species that you could work on? Of course, elephants have great memory, so probably an interesting species for hippocampal studies, but not very practical. So if you think of a practical small mammal, and I did want to work on mammals, about which quite a lot is known, maybe not so much about the neuroscience, but in terms of their natural behavior, bats come pretty much on top. And then they have also the various interesting properties that we've been exploiting over the years. They fly in 3 dimensions, so we can ask questions about how is 3-dimensional space represented in the brain. They fly fast, so you can ask questions where the speed of movement per se is important, and I can give you examples later on. They have 2 distal sensory systems, both vision and echolocation, so we can ask how is the same space represented by 2 different sensory systems that are distal, that are from afar. We don't have something like this, and rodents don't either, so that's an interesting advantage. We can measure the timing of their sensory inputs very precisely. By putting a microphone, we can record the timing of sensory input with a millisecond precision in freely moving animals. Again, something that's not so easy in some other freely moving animals. So there is a whole host of advantages to using bats. Of course, they're very social, etc. So I had a number of parallel reasons through which I eventually converged on this direction of going to bats. But this wasn't an easy choice because nobody worked on the hippocampus before in bats. So my postdoc was the first time anybody did that. So this was definitely a scary jump into the cold water to go in this direction.
Yeah. You said that grid cells hadn't been discovered then, but I guess by the time you did your postdoc, the 2005 paper, I think your postdoc was 2004 to 7, right?
Yeah.
Something like that. So they came out. Was that kind of also motivation for you to continue working in the field because the kind of obviously big things were happening in the field, or was it not even that obvious at the time when these things came out?
No, I think it was very obvious to anybody in the field that this is a big thing. At this point, I was already committed to this direction, so I don't think this changed anything, but it added pepper, so to speak. It was clear that there are more things to do. We can also look at grid cells now, which we did eventually. So that was definitely... and the funny thing is that I used to... I was working on the bat hippocampal recordings on the bat place cells, sort of in parallel to the Mosers and to Torkel Hafting and Marianne Finn, the 2 PhD students who did the work on the grid cells. And at SFN every year we would meet up in the SFN is a Society for Neuroscience annual meeting. We would meet every year in the alleyways of the sort of hippocampal posters, and we sort of used to have people asked us the same question. They sort of wanted both them and me. People wanted us to do like everything at once, do a study on remapping, manipulate this, manipulate that, and like we used to joke that you can do everything simultaneously. People have been working for decades on place cells, and they wanted me to do everything at once on bat hippocampus and them on rodent grid cells. So it was sort of a funny parallel. So I was very aware of this because I was talking to these guys a lot in real time during my postdoc.
You mentioned earlier that, bats have all these advantages for studying, but I don't know how much of a disadvantage it is, but to me it sounds like a massive disadvantage that maybe you could clarify kind of what the technical standards were at the time. Like, to me it seems like it would be harder to record from something flying around in 3D than from a rat moving around in a small box. Did you have to develop, for example, you have to store the data somewhere, and from what I imagine, usually that was done with a cable at the time, but I imagine that's not exactly ideal with bats. So you need some sort of way to transmit the data. And I'm curious, like, was all of that already in place in 2005, 6, 7, or whenever you exactly started working on this? Or was that did you have to develop all that technology to make it actually possible to study bats?
Yeah, no. We had to develop a lot of things. So even before, okay, let me restart. So we had to develop all of that. In my postdoc was on big brown bats. And then I moved back to Israel to work on Egyptian fruit bats, which are a lot bigger. And this is a big advantage. So during my postdoc, you had to start somewhere. So I did basically a comparative approach where I recorded in big brown bats while they were crawling in a two-dimensional arena. So sort of redid the rodent experiment, but doing it in bats. So, okay, I needed to set up tetrode recordings and solve some technical things, but there were still wired tetrode recordings. So this was very similar in many ways to rodent experiments. Okay, setting up for the first time in a new species, but nothing extraordinary, I would say. And the lab where I did my postdoc, the lab of Cindy Moss, they worked with these species, so I can learn from them surgical techniques, etc. So this was there was a lot to do, but this was not a completely new species or this flight issue that you mentioned. So this was something I definitely was able to do during my postdoc. And then when I moved back to Israel, let me go back for a second. During my postdoc, I was working also on developing, together with a company, Neuralinks, developing a wireless telemetry system for recording in the big brown bat. And this was an iterative process that took 3 years, pretty much, almost 3 years. Maybe 2 years, where we were working on developing this telemetry system, and it worked. The telemetry system was developed, went through several generations, but it ended up still being too big for the big brown bats. The big brown bats are called big, but they're really small. They're 15 to 18 grams. They're only called big because they're bigger than the little brown bat, which is 6 grams. So, big is in the eye of the beholder. So there was no way they're going to fly with this system. It was too big for the big brown bat back then. But then when I moved back to Israel and started my own lab and switched to the Egyptian fruit bats, which are 10 times bigger than the big brown bat, they could already carry this system. So sort of I used to tell people my low-tech solution to the miniaturization problem was to increase my animal tenfold. This is the useful way to go. So this is what I did, but still we continued miniaturizing things. So basically, when I started my lab, I took the system, the wireless telemetry neural recording system that we developed during my postdoc with Neuralinks, and applied it to the Egyptian fruit bats, and it worked very well because they could easily carry this weight. Michael Yartsev was my PhD student. He used this system that I developed in my postdoc to record from the bats, so it worked really well. And then later we switched to a different system, or developed a different system with an Israeli engineer, which is a different approach. Instead of transmitting the neural data, we switched to a neural logger system, or neural logging system that means storing the data on board the animal, which has three main advantages. One, it is unlimited in distance. So if we want to go to studying neural representation of very large spaces, then we are essentially unlimited in distance. The second thing is that it takes up much less battery power to store the data as opposed to transmitting it. So you can miniaturize battery and make everything smaller, which is a big advantage. And third, it also, whenever you're transmitting stuff, you're sensitive to noises, etc., interferences, which you can try to block, etc. But if you're recording right there on the animal, you don't have these issues at all. So it's also, by definition, a better quality of data. So these were like the three main advantages. So we switched to this neural logging system. And over the years with this company in Israel, Deuteron Technologies, we were developing already more or less 4 generations of these neural loggers that got smaller and smaller over the years. And now the 64-channel neural recording system that we're using now is smaller than the 16-channel system that we used, 6 or 7 years ago. So things have been miniaturized dramatically over the years. And there are other things that you need to solve, by the way. Just you're asking about introducing a new animal model. There are more basic things. For example, when I started working on the Egyptian fruit bats, we did not have stereotactic brain atlas. It's something that's very basic. Anybody working with rodents, monkeys, or standard animal models, they have an atlas of the brain so they can use it to direct recordings inside the brain. We did not have that, so we had to do our own atlas. It was 12 years of work, which a couple of years ago we published this as a book, but this was actually a lot of work to construct a stereotactic brain atlas. Then there are the things like anesthetic regimens that are different between species. You need to figure that out. This is not too difficult because, you played a little bit in 2 or 3 animals and then you converge on something. So there is a little bit of overhead with starting a new species. It is not that difficult in a way. The atlas is the main thing that you need to solve. The other things are pretty solvable. As long as you do electrophysiology, it's not an issue. The molecular methods that exist in mice, of course, are not easily transferable, or some of them are not easily transferable to other species. But as long as you do electrophysiology, you record electrical activity, it doesn't matter which kind of a brain it is. It's the same thing in a rat or a mouse or a bat.
Okay. You're mentioned a little bit one of the main advantages of the Egyptian fruit bat being that it's relatively large. I'm curious, was that the main reason, and that it also exists in Israel, or kind of why did you decide on that? Did you have a big process where there's, because there's so many species of bats, which one do I go with, or was it pretty clear that this was the only one that was going to work?
No, this was pretty clear that's the only one that's going to work for 2 main reasons. One is the size, which is on a technical level very important. And the second, it's also very common in Israel. So there's no problem of getting permits to capture them. Back then, they were even considered agricultural pests. Now, not anymore, but they're so common that it's not an issue at all. Whereas a lot of the other bat species are protected, so that's much more of a problem. With the Egyptian fruit bats, we're essentially unlimited in the numbers of animals that we can use. Not that we need big numbers. Through the entire lifetime of my lab, we've probably done experiments on the neural recording experiments. We've probably recorded, I think, on the order of 100 animals over all the last 15 years. It's small numbers, relatively speaking. But anyways, there's no problem to get permits with such a common species.
And so then how does it work? You just find a cave where they are and you bring a net and then you catch them yourself, or how do you get there? Because from what I understand, if you study something like mice or something, there's all these different kind of mice you can buy that have certain genetic properties, etc., etc. But I'm assuming that doesn't exist for an animal that is rarely studied in this way. So how do you actually just catch them yourself, or how do you get them?
Yeah, actually, that's exactly right. I catch them myself. So the way it works is I know several dozen caves or cave-like structures. These bats live in caves, and it's easiest to catch them in the cave during the day when they're resting. So I know several dozen places, and about 3 or 4 of them are relatively convenient for capture for practical reasons. The ceiling is relatively low, they're accessible, whatever. It's just these practical reasons. And yeah, we come during the day. I do the catching myself. We come, me and the vet, and usually another person. And, using a telescopic pole, for painting with a butterfly net, I just put it under the bats. So it's not, I don't, sort of scrape them, so to speak, off the ceiling. I just put gently the net under the bat, and the bat is trying to take off, but because of gravity, they're hanging on the ceiling. Because of gravity, when they take off, they sort of fall down, and they fall down just straight into the butterfly net. So it's very easy to capture several dozen bats within a few minutes like that. So, once or twice a year, we go and capture bats. It also means we are working with wild animals eventually. We also have a breeding colony in the Weizman Institute, where I'm based. But we've mostly over the years been using wild animals, which is another interesting advantage. We are studying the neural basis of navigation in animals that have actually navigating outdoors, that were caught as adults in the wild. Or if we are looking at social aspects in the brain, these are animals that have lived in colonies of thousands of animals, that had a lot of social interactions and were only caught as adults. So I think this is an interesting advantage as opposed to, inbred rodent species that could be different. It's hard to know, but in our case, we work on the wild animals, which I think is definitely an advantage.
I think in one of your papers, at least, you had this comparison between the wild bats and the bats that kind of grew up in the lab. So just briefly, just because you have that kind of comparison, kind of what kind of differences do you find in like, does it make a difference whether you have them born in the lab or caught in the wild, or are there some differences in their behavior and neural recordings?
So in behavior, yes, bats that are born in the lab that are used to humans from birth, they're much more friendly and maybe easy to train. There's some difference in the behavior. I mean, the wild bats are also very easy to train and to work with. Egyptian fruit bats are very, I would say, tame bats in a way. They're not aggressive at all. So it's very easy to work with them, very easy to train them. But the ones born in the lab are even more, more friendly. It's like, in a way, it's almost like a cat that was born on the street and grew up on the street versus the cat that was born in home. I think any cat owner like myself knows the difference. This is sort of a wild animal and this is not. So there is a difference in the behavior. In terms of the neural side, I think it's an interesting and important question. We have not explored it systematically. In this one study where we looked at this, what we did was actually, we asked a very general question. This was a study where we asked about the representation of very large-scale spaces. Maybe I should, before that, just to give a background about the types of neurons that we are studying to understand. So there are three main types of neurons that people have been studying in rodents over the years that relate to space. There are place cells, neurons in the hippocampus that whenever the rat or the mouse that are running in a sort of a small box, 1 meter in size or so, whenever the rat or the mouse are in a particular corner of the box or in the center of the box, in a particular location, the neuron will get activated, sort of in this one location. And this one location is called the place field of that neuron. And these neurons were called place cells by John O'Keefe, who discovered them in the early 70s. Then there are grid cells who are activated not in one location, but in a series of locations. Whenever a rat or a mouse passes through a series of locations that form a hexagonal lattice that span the environment. So place cells are thought to be like a cognitive map of where I am in space. Grid cells may be a cognitive distance meter, represent distances. And we also have cognitive compasses in the brain called head direction cells, neurons that represent not so much about, don't care about position, but they respond whenever the animal's head is pointing in a particular direction, sort of like a neural compass. So we have maps, distance meters, and compasses, all the elements that you'd like to build a navigation system in the brain. And basically, all of this was studied typically in animals moving in very small, empty environments. And what we've been asking over the years in the brain is how does real-life navigation differ from navigation in small empty boxes? And you can give several answers. One is that the world is three-dimensional, so how is three-dimensional space represented in the brain? And we've done quite a lot of work on that, which I'll tell you soon. You can ask, how do we navigate to goals? Because place cells tell you I am here, but how is the goal represented in the brain? You want to get to. It's a different question, and we've done work on that as well. Third answer is that we often navigate in groups. Animals often navigate in groups. And so how is the social factors come into play? And we'll talk about the social aspects later. And lastly is the issue of spatial scale. Of course, normally in real life, humans or animals do not navigate in 1 meter boxes. We navigate in much larger spaces. And of course, nothing was known about how our very large space is represented in the brain. So to do this, we constructed a very large tunnel, a 200-meter long tunnel at the Weizman Institute. Now it's 700 meters long. And we developed those neural loggers that I told you about before, these wireless recording devices that store the data on board the animal. So now we are unlimited in distance. And now we can record and, we measure the position of the animal using not a GPS system, something more, about 10 or 100 times more precise than GPS, where you measure distances from a tag on the animal, a radio frequency-based tag on the animal, to an array of antennas that we position around this tunnel. So we can measure the position of the animal with about 5 centimeter precision nowadays. And so we measure the position of the bat, the neural activity, and now we can look at place cells in the hippocampus in these large environments. And what we found, and this was published in Science a couple of years ago, unlike in small environments where neurons are active in one location, so sort of these place fields that I mentioned are sort of Gaussian small place fields in one location, in this very large environments, each neuron was active in many locations. And surprisingly, these many place fields of one neuron could vary a lot in their size. So the same neuron could have a 1 meter place field in one location and a 20 meter place field in a different location. So it's sort of a multi-scale or multi-resolution type of a neural code. And we had also a computational, a theoretical component in this study, and we showed that this type of a multi-field, multi-scale neural code is much better, 2 or 3 orders of magnitude better than the classical place code in terms of decoding accuracy. And when we saw this very surprising type of a multi-field, multi-scale code, we also looked at whether in lab-born bats we'll find the same, because this was all done in wild-born bats. And then a very natural question to ask is, if you take a lab-born bat that has never seen hundreds of meters in his life before, will we find the same thing? We thought that we will find something different, and very surprisingly, or even shockingly for us, we found the same type of neural code also in the lab-born bats. So exactly like in the wild-born bats, we found this multi-field, multi-scale code also in the brain of lab-born bats with very similar characteristics. So this suggests that this is an innate, this type of a multi-field, multi-scale code is an innate property that's very fundamental to the function of the hippocampus, regardless of experience.
Yeah, it's a really cool study. And I mean, just from a practical perspective, how do you go about building a 200 or now 700 meter long tunnel? I'm assuming you're not digging underground for a tunnel. It's just so bats have a tunnel to fly through. But I mean, I'm curious. Yeah, it's just like, how do you, because I guess you need, usual neuroscience labs aren't 700 meters long. So I guess you have to have some sort of large area of land where you can actually do this and then actually make it happen. So I'm curious whether you could comment just on the practicalities of kind of setting up this very unusual kind of lab.
Yeah, no, it's a great question. Indeed, it's not trivial at all. So just to understand sort of the rationale, because this will come in the story for a second, the rationale for doing this study was that if we want to sort of, we did early on in 2010, 2011, we did a study with GPS tracking of bats, where we put tiny GPS data loggers and tracked the navigation of bats outdoors, just a behavioral study. And we found that these Egyptian fruit bats have these very nice flyways where they fly from their cave to a particular fruit tree night after night, very precisely. So they can fly 15 kilometers to the exact same fruit tree night after night. So they are really good navigators. But if you look now at those flyways, what is the volume of the flyway? It could be, for example, if a particular animal flies, let's say 30 kilometers, so the length of the flyway is 30 kilometers. The width could be about 2 kilometers. So like the variability between days could be up to 2 kilometers. And in the altitude, the variability is about half a kilometer. So we have like a sausage at 30 kilometer long, 2 kilometer wide, half a kilometer high. That's about 30 cubic kilometers. That's the volume that you need to represent. And if you now want to take your 10 centimeter size place fields that you measure in small laboratory boxes and use those place fields to tile 30 cubic kilometers, then you can make the back of the envelope calculation. And you also want some overlap for robustness. It ends up being that you will need about 10 to the 15 neurons for that. So 1,000,000 billion neurons, which is a lot more than what you have in the hippocampus, because the entire dorsal hippocampus C1, this particular area where place fields are found, is about 10 to the 5 neurons, or 100,000 neurons in bats or rats. That's the order of magnitude, okay? Maybe 2 or 3 times more than that, but that's sort of the order of magnitude. So 10 to the 5, not 10 to the 15. So we have a gap of 10 orders of magnitude. This is an enormous gap. This means that what we measure in small laboratory environments cannot translate to what's going on outdoors. Just impossible. There must be something else. This was sort of the rationale of how we came to do this study in the first place. But this was also the rationale of how I got this tunnel built. Because as you say, this tunnel, it's not underground, it's overground. It's more like a glorified tent, a long, very long tent, so to speak. A very robust one, but it's sort of a tense lightweight structure, like a greenhouse or a tent, if you will. And what happened is that early on, I had this idea that, it's very important to record in larger spaces if you're studying the neurobiology of navigation. But how do you do that? There's no space for that in buildings, right? So I went to the president of the Weizman Institute back then, Daniel Zeifman, and I gave them the spiel of why is it important. And the spiel was exactly what I just told you about this 10 orders of magnitude gap. And, the president, he was a physicist, so he doesn't know much about the brain, but he knows about 10 orders of magnitude. So when you tell a physicist who have a problem that's 10 orders of magnitude large, he understands that very well. Okay, that's a big problem. This is something worthwhile pursuing. So after I gave him this spiel, and so he asked me, okay, so what exactly do you have in mind? How do you think to come about? What do you want to do exactly? I told him, I think the solution is to build a lightweight tunnel, like a long, lightweight construction in some empty area on campus. And this was probably the moment that I had the highest heartbeat in my life. Probably I like 200 or whatever, 250 beats per second, because I was sure he will just kick me out. But what did I have to lose? So I gave him the spiel. I told him that's what I think we need to do. And happily for me, he said, he rose up, and I was sitting in his office, and on the office of the president there was this huge, like the whole wall was covered by an aerial photo of the institute, few meters in size, and you sort of stood up, came up to this photo, huge photo, and sort of looked at it and said, okay, let's see, where do we have space? And I yes. So when there is a will, there's a way. And, he did find space at the end of campus, which is also good because it's an area that nobody wants to build a building at the very far outskirts of campus. Scientists want to be, near their friends, near where the lectures are, near where academic activity is. So actually being 5 or 7 minutes drive away at the very edge of campus is very good. Nobody wants this area. We do need to commute there, so we had to buy a golf cart, so we have a laboratory golf cart for, taking, because the bats don't live there in the tunnel. They live in their lab, and for the experiments, we take them there, we do the experiment, and we drive back. So we have a golf cart for this driving, but, this has ended up being a very convenient arrangement.
Yeah, that's an unusual bill for a neuroscience lab to have a 700-meter long tent and a golf cart.
Yes, exactly.
But hey, I guess. But I mean, that's cool that, he kind of saw the problem and just kind of, I guess, trusted you that your solution also works for it, right? As you said, he could it seems like the more defendable decision would be to say, can't you just do normal neuroscience? Do you have to do this? But yeah, it's pretty cool that he did that.
Yeah, I totally agree. I think I really appreciate that. It's not obvious, but I think good managers, that's what good science managers, that's what's supposed to do. I need to trust their scientists, and just, as long as it's feasible, just, go with it.
And so then with the 700-meter tunnel, it was what do you gain from the extra 500 meters there in terms of what kind of scientific questions can you answer that you couldn't do before?
That was, the original tunnel was a sort of quick construction. We did not have air conditioning. We did not have a lot of things. Now we sort of added air conditioning, which makes a big difference, because if you're trying to run an experiment in a greenhouse in August in Israel, good luck with that. So students ended up running experiments in the summer between 6 a.m. and 8 a.m. in the morning, and then starting at 5 in the afternoon and until midnight, or sometimes even 2 in the morning. So it was totally crazy hours. Actually, 8 to 5, which is the more normal working hours for most people, we could not run experiments in the summer because it was too hot. So now we actually can, because it's air conditioning. This is a practical thing. Also, we added some turns, so we have now an ability to have decision making. The bat has to decide whether to continue straight or turn right. We also built a very large flight maze, 60 by 35 meters, so sort of almost a football field size maze, which is like a real proper maze where we can do complex navigation tasks. and ask many questions that are related to navigation, as opposed to flying back and forth in the tunnel, which is great for asking questions about the representation of space, but it's not really challenging navigation in any way. And whereas in the maze is challenging and interesting. So we've added a lot of components now with this new construction, but the basic idea still remained the same.
So how does this cost a lot to build this stuff? I have no idea what even order of magnitude we're talking about.
It's millions, and it did require a donor to give money for that. But again, it's we did found a donor that was very interested in that, so that ended up being fine.
Yeah, you mentioned earlier something that surprised when you talked about the kind of the distances that the bats travel, and to some extent was it 20 kilometers, 30 kilometers? I've forgotten now.
Yeah.
But that large distance to some extent surprises me less than the 500 meters height. What are they doing in 500 meters height? I mean, supposedly there's no cave there, there's no tree, there's no are there flies there? I don't think so, right? Or what are they doing that high?
Right, so they fly high only during commuting. So when they around the cave, they fly low. When they're around the fruit trees where they eat, they also fly low. But when they're doing these straight, long distance commuting flights from point A to point B, they really go up high and fly very high. And I think the reason is because they're very visual animals. It's one thing that important to mention, that although bats are about almost 25% of mammalian species. So there are about 1,400 species of bats in the world. So about a quarter of mammals are bats, and they all see. Some see not so well, but some see very well. Our bat species have excellent vision, much better than rats or mice. So they're very visual. They also have the sense of echolocation, and because they're so visual, when you fly high up, you can see far away, and it's better for navigation. And we've, in fact, in this GPS study that we've done some 12 or 13 years ago, we've done an experiment where we translocated the bats to the desert in Israel and released them inside this crater, inside this deep machtesh, this deep crater, from which they could not see their familiar area, and then they sort of got lost. They were flying around and around and around until they flew higher up and could see the familiar area, and then they started flying straight. It was very clear that once they see the familiar area, they start flying straight. So they do rely on vision for navigation. Probably they use also other senses, but vision is very important for long distance navigation, and then when you're flying high up, you just see further.
Yeah. I mean, I was wondering about that when you talked about these large distances, and correct me if I'm wrong, but echolocation needs something to bounce off of, right? So you're very limited to, I don't know how far that goes, but you can't, you can look like 20 kilometers far or something depending on weather conditions, right? But
right, yeah, you're right. Echolocation, when you're flying at an altitude of 500 meters, echolocation will not work at all. The furthest that you can get echoes from is from a few tens of meters, maybe 50 meters from the ground. But at these altitudes, you don't get echoes from the ground at all. You might get an echo from another bat or something like that, but not from the ground. So it's basically useless for navigation to fly at such altitudes. And in fact, there have been experiments, a study in the 1960s, something that ethically will not be approved today, but where essentially they looked at how bats, it was done in the US in insectivorous bats, not in our species, but normally these bats fly relatively high when commuting, but they actually caught some bats and took out their eyes. Okay, so not today, it will not be approved, but in the 60s people did such experiments, and then they describe in this paper that the bats flew much lower, as if they were sort of palpating the ground with echolocation, because now they had to rely on echolocation to navigate. And so, yeah, if you use only echolocation, you have to fly low for this to be useful.
Yeah, and I guess you also then I imagine it's harder to not see the forest for the trees or whatever, right? Because you can only look at the very short distance stuff, so you lose this kind of larger perspective to some extent. So I'd imagine it's
exactly
it also limits you in terms of not getting lost over large distances.
Exactly, yeah. You can't see far away landmarks like mountain ranges or the sea or cities or any of these far away things that are kilometers away. You can use vision for this, and we think the bats do use that, but with echolocation, it's all very local. So it's a completely different mode of navigation. And that's something we have to recognize generally in neuroscience, that different animals have different sensory systems. And even the same animal, when it uses different sensory systems, its worldview is completely different. The same animal, the bat, what it can use in terms of information when it's using vision versus when it's using echolocation, it's a completely different thing. And this is something that I think by studying non-standard animal species, you become more aware of this diversity.
Yeah, I mean, I think you mentioned there are some, on your website, you have some links to some videos and like short documentaries about your lab. And I think in one of them you mentioned this, that, you can use these two different modalities of sensing the environment, let's just say, with echolocation and vision, and separate them and kind of see like, how that changes their perception and their navigation, that kind of stuff. And obviously you can't do that in other species, and that's kind of also one of the main advantages of using bats. Right.
Yeah. We've done some years ago a study where we asked how is the same space represented using vision versus echolocation. So we had bats either like fly between two landing balls, either using echolocation alone, so in complete darkness. We had to use night vision goggles to orient around, or using vision only, and we blocked echolocation by transmitting broadband sound through some speakers, and we add separate control to make sure that the noise itself did not have an effect. And we also had ventilators to blow away any odor trails in midair, if such they do exist. So really to create a clean double dissociation between vision alone versus echolocation alone. We worked hard to achieve this double dissociation. And then what we found is that the same space is represented using two different maps. So place cells in the hippocampus represented space both using vision and using and when using echolocation, but the locations of the place fields moved around, or in some neurons they shut off or place fields appeared. So there was what's called a remapping, a new map or a different map between when you use vision alone versus when you use echolocation alone. And this goes against the original idea of the cognitive map that was proposed to be an abstract concept, right? If you have an abstract map of space, it shouldn't matter whether you use vision or echolocation. But in fact, if you think about it, when you navigate in daylight or at night in the same space outdoors, things look pretty different. So turns out that in the hippocampus, at least, the maps are different. Now, of course, there could be still an abstract map elsewhere, for example, in entorhinal cortex or somewhere else, but at least in the hippocampus, the map is not entirely abstract, but does depend on the sensory system. And this is something that we could do by using the bat as an animal model, but it's hard to ask the same question in rodents, for example, because they don't have two distal sensory systems like the bats do.
On this topic of like different species have different advantages, is there some species that you think is kind of being overlooked in spatial navigation? I don't know, something where you're like, I wish I could do this thing and that species could do it. I'm just curious whether there is anything like that.
Yeah, I think one general, it's not a particularly specific species, but a class of species I would say that we are missing is migrating species. So there is work being done on understanding migration, but that's mostly at the behavioral level, and a little bit people are looking in the brain, but it's more looking at magnetoreception in birds, things like that, not really looking at individual neurons. But, migrating species are amazing animals because first of all, they fly thousands of kilometers. That's now a completely different scale. Our bats are non-migratory, so they fly a few tens of kilometers. That's a lot compared to a lab, but it's much less than some of the amazing migrating species that can fly thousands of kilometers. By the way, there are some bat species that are migrating 2,000 or 2,500 kilometers. So some mammals do that. Then there you could ask questions about navigation. First of all, how do you navigate such large distances in such a precise way? Also about the timing. How does the animal decide that, today is my time to take off from Africa and to go to Europe, or the other way around, right? I mean, these are, I think, fundamental questions that we have very little understanding how these things works in the brain. So I think this is a whole domain that we have no knowledge about. So in terms of, I would say, the missing types of animals, it would be migrating animals.
Okay, that requires a very long tube.
Yes.
But, yeah. I have two kind of random questions about bats, just from not really, like, from personal experience a little bit. I've just always wondered about, but don't know the answer. I'm curious whether you happen to know it. So basically, where I grew up, which is like in Germany, on the border to Netherlands and Belgium, there are bats. I can't find out which species because, as you said, there are so many. So like even in Germany, there are like 30 species of bats or something. But one thing I always find it kind of interesting is that the only way I really see them is like I'll be walking along a road when it's dark, then you come by a lamppost and then you see something flying around there. And my initial assumption is always like, oh, it's, some bird or whatever. Because bats are not like, you forget that they existed because you never see them, apart from that one situation. But then when you only see them like flying underneath a lamppost, I'm assuming getting some mosquito or something like that. You only see them for like half a second, but in that half a second, it's enough to realize that thing that you thought was a bird flew really weirdly, because they have this like, much more angular way of flying. I don't know whether that's the right way to put it, but like, it's very obvious that it's not a bird just from the way they're flying. So I'm just curious, like, why is the way that bats fly so distinctive that I, as someone who's basically never seen a bat in my life, can immediately tell that that's not a bird, but a bat?
So there are two basic answers here why the flight of bats looks different. One is size. So a lot of the bat species are very small, few grams or 10 grams.
Small.
And the really small species actually fly almost like a large moth. So it looks sort of erratic, and it has to do just with their size. So one thing, and there are almost no birds of this size, right? So birds are generally bigger, most of them. So this is one difference. So bats are just, a lot of them are very small. So this is one answer. And the other answer is that their mechanism of wing flapping is different from the birds. The birds have this joint that's different than in the bats. And, there actually are people who are studying aerodynamics of flight in birds versus bats, and bats are different. They can achieve the same end goals. So there are bats that can hover in one location, very similar to hummingbirds.
Really?
Yeah, for nectar feeding bats, for example, can hover in one location to drink the nectar of flowers. So the end result is similar, but the mechanics of the wing is quite different. So that also explains why flight in bats looks different. Yeah, bats are very varied. You're surprised by the fact that they eat nectar, but there are a variety of species and variety of foods that bats eat. There are of course the insect eating bats, but there are the fruit bats like the ones we are studying. There are nectar drinking bats. Baobabs, for example, pollinated by bats. Agave cactus in Mexico, pollinated exclusively by bats. Agave is used for tequila. So we bat researchers always say, no bats, no tequila. So bats pollinate some important plants. There are bats that hunt small animals like frogs. There are bats that hunt fish underwater. Amazing bats in Central America that will detect a fish that comes close to the surface of the water. It creates a circular ripples. They can't, of course, detect the fish itself, but they see in quote unquote see. They see with echolocation the circle created by the fish, the ripples, and then they put their claws in the middle of the circle and take out the fish. So the bats that do that, and then there are of course the vampire bats, 3 species of bats in Central and South America that feed exclusively on blood. So they're really very diverse in what they eat.
Yeah. One thing that really surprised me, just because I looked a bit more at bats before we started speaking, is just these enormous bats that really, they look scary. I'm glad I don't see one of those flying around. I don't know. I think those are fruit-eating bats, but
yeah,
the ones that I saw. But yeah, they can get big.
Yeah, some of the bats that live in some Pacific islands or in Australia or in other places can get to almost 2 meters in size, 180 meters wingspan. Our bats are actually from the same family, so small cousins of those fruit-eating bats. Same family, different genus.
Okay. So the other random question I had is that I guess I'm kind of curious, kind of in part where bats live, because the one thing that's really surprising here is that, like, as I said, you don't really know that bats live here until you occasionally see them in the evening. And I have no idea where they live. I mean, do bats, the stereotype, I guess, that they live in a cave, right, and hang from the ceiling. But do they also, I don't know, do they just live in trees, or do they some build nests, or or could it be that these, bats like you described actually live like 10 kilometers away or something, and, some small cave somewhere?
No, it's a great question. The bats actually are very, just like they're diverse in the food items that they eat, they're also very diverse in the type of roost where they live. So there are, on one end, the bats that live in caves. There are many species that live in caves, but then there are bats that live in little rock crevices, or that live in tree hollows, inside hollow trees, or bats that live under a bark of trees, or even bats that create nests, so to speak, not exactly like bird nests, but there are these tent building bats in Central America that will sort of chew on these big leaves. Think of like banana leaf, but those sort of types of leaves that are very big, they can sort of chew on the central stem of that to make them sort of fold and create a little tent, and then they would sleep underneath this tent. So that, of course, will hold for only a few days. So, of course, if you live in a cave, you tend to live there for many years. And then if you're creating those tents, you have to create a new one every few days. And by the way, I didn't mention, but bats also live very long. Unlike a mouse or a rat that is old at the age of 1 or 2 or 3 years, bats can live dozens of years. There is a bat species that's 10 grams in size that was shown to live up to 47 years. There was one individual that was recaptured in the wild after 47 years. Our species live at least 25 years. We know that, maybe 30 years or more. So they live very long lives. That also means that they have many years to learn the layout of the land and to learn to navigate very well in this area where they're living.
Yeah, no, yeah, as you said exactly, like, I assumed that they'd live, more or less like a rodent in terms of duration, but okay, I had no idea. Yeah, it's still kind of, I'd like to know where these bats live. I don't know, maybe I can find like some local, like...
Yeah, so it's hard to, it depends on the species. The big brown bats, for example, that I worked on in my postdoc, nowadays they love to roost in attics of people. It's a great place. People don't go to the attic for years sometimes. And so, it's a convenient place to live in, quiet and heated, protected. So attics have become very popular amongst this bat species. So, it really depends on the species.
Yeah. I'm always fascinated with, I mean, I grew up in a fairly rural area, but like how much, wildlife there is once you actually start paying attention to it. Even in places where you wouldn't assume it. I mean, yeah, I don't know, maybe this is specific to where I grew up, but like we even have like beavers. I didn't know we had beavers here until I suddenly like walked through this one area where there was like a river, and suddenly you see all these like trees with the gnawed off stump of a tree.
Yeah, we have actually on the Weizman campus we have quite a lot of wildlife, because we have like a fence around the campus, so it's protected area, so nobody bothers the animals. And actually close to the tunnel, as I told you, this tunnel and maze that we have are at the end of campus, and there's like a big empty area there. It's like a little piece of wilderness, so to speak. It's close to a road essentially, but people don't go there except us, basically. So there are some jackals that live there. There are some foxes. There are some pretty rare bird species that you can see there right near our tunnel. It's pretty crazy.
Yeah, it makes me just want to go and go a bit more into the forest and just watch, just kind of just stay there and see like all the stuff you can see there. Anyway, going back to kind of your research on bats, I guess one thing I wanted to talk about a little bit more is the social lives and the social aspects social lives of bats and the social aspects that you studied. Maybe to kind of start that conversation, again, huge difference between different species of bats, but maybe for the Egyptian fruit bats, kind of what is their social structure? Do they, I saw, God, I mean, this is a different species, but there's this one like cave in the US where there's like millions of bats. It's crazy video. But kind of what's the social structure of those bats that you use? Do they, live in large flocks? Is that the word? I don't know. Or kind of what's that like?
So they live in colonies that are not millions, but between a few tens and a few thousands. That's sort of the typical size for this species. It actually, again, varies a lot between different species. There are bat species that are solitary, that live alone. There are bat species on the other extreme. There's Brazilian free-tailed bats in Texas and New Mexico that you've probably seen videos of, that have millions of bats in one cave. And then there are all sorts of things in between. So different species differ a lot in the numbers and in the social structure. Embarrassingly enough, we don't know a lot about the social structure of our bats. We know, it's not a pair structure like pair bonding for life. It's more of a fluid thing, and they have a hierarchy. They have dominant males and females, less dominant ones. So likely the more dominant ones are the ones reproducing, but exactly the details of the structure, we don't know that much. But it's sort of a dominant type of structure.
Do they form friendships? Do you know anything about that? Like where you realize, those two are always hanging out together? Or?
Yeah, they do. And actually, we've looked at that in a recent study. They have preferences for interact, with particular bats and not with others. And some of the reasons that we went to look at social aspects have to do with the representation of space, but some have to do with the social questions per se. So it started from, again, several directions. We were looking at representation of space and neurobiology of navigation, but then, one of the things you need to do is to train bats to do all sorts of tasks. And one of the things that we found very quickly is that if you use one animal as a teacher, you found one animal that does the task, whatever the task is, does the task very well, and just let it perform the task while other bats watch it, they learn the task much faster than from a human. So a bat trainer is much better than a human trainer. And so there's a lot of observational learning going on, much more than in rodents. And so for many years, we used it as a training trick, but then we actually at some point realized that this is an interesting question in its own right, scientifically. The other reason is that we studied the hippocampus, this brain area where you have place cells, neurons that represent where I am in space. But then for such a highly social species, you can ask, are there also neurons in the hippocampus that represent where you are in space, where another individual is in space? So this was a study that, the first study that we did on the social aspect, looked exactly at this question. We did a task which involved two bats, a teacher or a demonstrator and an observer bat, a student, so to speak. And this was a mimicry task. The observer had to mimic the behavior of the demonstrator, mostly so that it would pay attention to the position of the other bat. This was the main rationale here. And what we found is that while the bat itself, the recorded bat, was flying, we would see place cells, normal-looking place cells. When I'm moving through space, I have neurons in my hippocampus that are active when I'm in a particular location in space. But when I was resting and you were flying, so the other bat was flying, we found that there are neurons that were activating whenever the other bat flew through a particular location in space. So I'm not moving at all. And we had actually a motion sensor on the head, an accelerometer, to verify that the recorded bat was not moving at all and was just watching the other bat. And still, the neurons in the hippocampus represented the position of the other bat. And we call these neurons social place cells. And, we had all sorts of controls to show it can't be explained by all sorts of other explanations, and that it's sort of, there was a social aspect here because we also moved plastic objects through space, and the neurons responded differently to the real bat and to the plastic objects. So there seemed to be something really social here. So we call these neurons social place cells, neurons that represent the position of another individual, which is, of course, very important for all social animals. To know where is somebody else is very important. My kids are grown up now, but when they were little, we'd go to the park and you always pay attention, where's the little kid so it doesn't run to the street or something, or where is your spouse or your friends in a bar or whatever. So paying attention to the positions of other individuals is important. Or in any sports games like football, basketball, things like that, the really good players are ones that, of course, in addition to the physical sides of things, but the really good players are very good at always keeping track of where are the other players on their team and the other team. So this ability to track the positions of other individuals is very important for social animals, and these social place cells might be related to that. And then after that, we sort of asked the next question, sort of an obvious question, which is, instead of having just one other animal, what happens if we have multiple other animals? How do you represent them? And so we created this. So the first study was already published a few years ago in Science, and what I'm going to tell you now is an unpublished study that we're just submitted. And what we're seeing is that when we create this colony of bats in the lab, so sort of a group of bats, and we develop methods to track the positions and identities of all the bats in the groups of about 5 to 10 bats, we found that there are neurons that represent the positions of all these other individuals, but they also represent not just the positions, but they also represent, we're sensitive to presence or absence of another bat, and also to the identity of another individual. Is it one bat or another bat that's in a particular location? And also, these neurons represented the other features of the kind of important social features, like the sex. They responded differently to males versus females. They responded differently to, depending on the dominance hierarchy. Is it the bat high in hierarchy versus low in hierarchy? And also, they responded differently to, are you my friend or are you not my friend? And we saw, you track behaviorally in this colony, we see that they have preferences. You can identify who is my friend because, they tend to interact with one individual more than with another. And these preferences are stable over many weeks. So it's really sort of friendships, if you will. And these friendships were represented by the neurons. So there's a lot of very strong social signals that we see in the hippocampus in this highly social group situation.
Mm. I'm especially wondering, like, as you said, like if you have like multiple other people or bats or whatever in the same room, do they track all of them or just the kind of ones that are relevant for a particular task or that kind of thing? Because I don't know, I'm curious, like, especially what you mentioned, for example, with people, like the example football, if you're a midfielder, you might have like 10 people around you, right? Yeah, I would imagine it's difficult to keep track of all of them and where all of them exactly are moving as individuals and that kind of stuff. So I'm curious whether you, especially those that, maybe aren't relevant for what you're trying to do. So I'm just curious whether you differentiated there between how relevant kind of a particular bat was for a particular task.
Yeah, so that's a great question. In this experiment that we did recently with between 5 to 10 bats, we found that neurons did tend to represent the individual. So not all neurons. Some neurons tended to generalize across individuals or only represented a few of the individuals, but some of the neurons represented all the various different individuals. So if you combine all the neurons together, you can tell apart which individual is that. But that's for a relatively small group of 5 to 10 animals. What happens if you go to a whole colony of thousands of animals, or to those colonies with the Brazilian free-tailed bats where you have millions? I mean, you don't even know millions of people, right? You can't represent them individually. Yeah. So I agree with you that it doesn't make sense that all individuals are represented. It's probably by relevance. So if you go to a full colony of bats, I think, but this is an open question, it's something that needs to be done, with much larger groups, is that, either you'd represent just the, let's say, most important animals, like the animals highest in the hierarchy, for example, or the ones most relevant, for example, the ones closest to you. You don't care about the ones far away, only the ones closest to you, or the ones that are on collision course with you, or other criteria of relevance. But I agree with you that that's the most likely situation. I don't think that it makes sense for the brain to represent 10,000 animals simultaneously. I don't think it's feasible, and probably that doesn't happen. But it's an open question. To show that, it's a different thing.
Yeah, I mean, I guess some of it, I assume, would happen also at the sensory level. Like I was just thinking, like if you enter a subway or something like that, as in underground tube, not the food place, then I guess even in the food place, there are maybe some people who are sitting like 5 meters away from you at a bench. You probably don't even perceive them, but I guess the closer they are to you and that kind of stuff, you perceive them, and then there's some people you ignore anyway. But yeah,
right. And I think that's another reason why we need to move to larger spaces for neuroscience research, right? So it's maybe not the most important reason, but what you just said is that in a large enough space, I care about the individuals close to me and not far away. But if you only do experiments in very small spaces, then all individuals are close to you. So you'd think that the neurons care about everybody. In this particular experiment, it was a relatively small space. It was a room of about 3 by 3 meters in size. And when most of our analysis were done when the animals were really close to each other. So everybody is relevant. But you're right that in a very large space, this could be very different. So I think for that, you need to do the experiment in the large space. So just like it's important to go to large spaces to study the neurobiology of navigation, even for social questions, it's important to go to large spaces, not maybe 700 meters, but larger than normal.
Yeah. I have kind of one fairly general spatial navigation question, which especially to me, and yeah, it's kind of a question I have increasingly when I kind of read new studies. The question is kind of like what to make of there being so many different cells, place cells, grid cells, that kind of stuff, and that so many of them are modulated by so many kind of contextual factors. And kind of what I mean by that is that, when you start off, learning about this stuff, you learn here's place cells and there's grid cells, head direction cells, and then, you get to border cells and all that kind of stuff. And it just increases, right? You just learn it seems like, every other paper in spatial navigation has a new type of cell. And I'm not entirely sure what to make of that, but at some point I wonder, is this a sign that we actually don't understand the underlying principles? Or is it just what happens naturally if you have animals or people moving in complex environments that you need to take into account all the complexities of the environment? I'm just curious whether you could kind of comment on that, because it's just something I'm increasingly wondering about without really knowing exactly kind of what my question is there.
No, it's a great question, but I think actually it's two different questions that are involved there. One is the issue of, are there distinct cell types? Because in simple environments, we tend to see these simple neurons, grid cells, place cells, head direction cells, etc. And we tend to classify them. And in more complex environments, things become more complicated. For example, what I told you about this long tunnel where a place cell in the hippocampus has multiple place fields, that starts to sound a little bit not so different than grid cells that also have multiple place fields that are maybe more periodic. But, so things become more, a little less distinct when you go to more naturalistic situations. So the question of, is there a really distinct sense of set of neurons, or is it more of a continuum? I think it's an open question. There is some evidence that grid cells maybe are a distinct set of neurons, but that's also an open question, I think. But for a lot of the other cell types, I think we need to understand, do you have complete mixed selectivity where all neurons represent sort of everything? And when you go to very simple laboratory setups, then you end up seeing the extremes that neurons represent this or that, and then you categorize them into cell classes. But maybe in more naturalistic situations, things are more of a continuum. This is sort of one question about cell classes. The other, and very related question, is about naturalistic or natural environments. Because for the same type of cell, what we have been showing over the years is that for the same type of cell, when you introduce more naturalistic aspects, you find that they have more complex responses. So when we looked at 3 dimensions, we found 3D place cells, neurons that represent the 3-dimensional position of the animal. We found 3D grid cells that we didn't talk about them yet, but unlike the 2-dimensional grid cells that have this hexagonal pattern of activity in space, in 3 dimensions, they have a local distance metric between their grid fields, but not a global lattice. So this sort of quite different. The grid cells look quite different in 3 dimensions than in 2 dimensions, which have important implications for theories of grid cells. We looked at 3D head direction cells. We found these interesting properties. We did find 3D compasses, the 3D head direction cells, but they were represented on a toroidal reference frame, not in a spherical reference frame. So some interesting findings there. Then when we introduced a navigational goal, instead of looking at where I am in space to look at how is the goal represented in the brain, we found that we found vectors in the brain. We found neurons in the hippocampus that represent the egocentric direction and distance to the goal. So literally a vector to the goal, which is very useful for navigating to goals. So a very different type of representation than place cells. Then when we introduced other animals, as I told you, we found those social place cells, neurons that represent the positions of other individuals and sex and dominance hierarchy and affiliation, etc. A lot of very strong social signals. And when we increase environment size, we found, all hell breaks loose. We find this multi-field, multi-scale type of code. So every time we take a step into a more naturalistic direction, we find something more complex and interesting about the brain. First of all, I think it's really important. It shows that it's important to study the brain under more natural conditions. And I'm now actually finishing writing a book for MIT Press called Natural Neuroscience. I'm submitting the final version of the book actually in a few weeks, so it's almost done. And the idea of the book is exactly to talk about why is it important to study the brain under natural conditions. Another aspect of your question is everything represented in the neuron? So is anything that you throw at the neuron will represent? And I think the answer is not everything, but maybe in the hippocampus at least, the answer is that everything that's relevant. And the reason I'm saying this is because, as I just told you, we have these social place cells, neurons that represent the position of other individuals in a social situation. But we also did an experiment in the tunnel where we had two bats flying opposite each other. This was a paper in Nature last year. And what we found there is that when the bat is flying alone, it's representing its own position with this multi-field, multi-scale code that I told you about. But when they're flying opposite each other, they're switching between representing my own position to representing the position and the distance to the other bat. And when they're passing each other, the neurons are going back to representing just my own position. So there's this switch from representing my own position to representing position and distance, and then back to representing my own position. So this was sort of the reason to do this study, was to look at how very dynamic type of behavior, or when you switch behavior from navigation to collision avoidance, how does that affect the neural code? And the answer is that it affects the neural code dramatically. The neural code is very dynamic when the behavior changes dramatically. But one interesting aspect of that study is that we did a variation of the experiment where instead of having one other animal, we actually switched the other animal. So instead of bat A flying opposite bat B, we had bat A flying opposite bat B or bat C. So we can now compare what happens when bat B or bat C is flying opposite me. And it turned out that there was actually no difference. The neurons switched to representing the other bat in exactly the same way. It didn't matter if the other bat was bat B or bat C. But it actually makes sense because this is not a social situation. It's a collision avoidance. When you're driving on a road and you're about to collide with another car, you don't care if this car or the other car. You just mostly care getting the hell out of there and not colliding, right? So it's not a social question. And indeed, the identity in this condition was not represented. Whereas in the more social situation, the bad identity was represented because it was relevant. In a social situation, bad identity is relevant. In the collision avoidance situation, bad identity is irrelevant, and then it's not represented in the neuron. So I really think, and we have several other examples like this, where if something is relevant, it's represented, but if it's irrelevant, it's not represented by the neurons.
I know you didn't call them collision avoidance cells, but I think that would be a nice addition to the list of cells. Yeah, it's funny. I wanted to ask you a little bit about how the natural neuroscience book is going, because I saw on YouTube, I saw a talk you gave a couple of years ago or so, where you mentioned you were writing it. So I was like, how's it going? So, but I guess it's progressed quite a lot then if you're about to submit. Do you want to say a little bit more kind of about this? I think in the same talk you talked about a kind of slightly false dichotomy of controlled but unnatural and versus controlled but natural. Yeah, do you want to elaborate on that point?
Yeah, I think it's sort of the main point of the book, the Natural Neuroscience book, is about the importance of going to more naturalistic behaviors. And the basic idea is that we have typical experiments in the lab, especially in the olden days, have been very controlled, but also very unnatural. And I always fear that if you do very unnatural experiments, especially on the neural base of behavior, it's a problem because we might be missing important aspects of what the brain is doing. We don't really let the brain do its normal thing. On the other extreme, you can do something that's completely uncontrolled but completely natural, and that's hard to analyze. It's not a lost cause, because often if you let animals do whatever they want, they would still do some behaviors repeatedly. Like in our colony situation, they would fight over and over. They would share food over and over. They would repeat certain behaviors over and over, so you can still align your neural activity on these repeating behaviors and see how the responses look like. And it's not a completely random behavior. So even if you don't have any task and you let animals do their completely natural behavior, you can make sense of the data, and we are showing that with our studies. But my point is that it's important to have a diversity. So between the one extreme of having highly controlled but highly unnatural studies and the other extreme of highly uncontrolled but natural studies, first of all, I think we as neuroscientists need to cover the entire range. And the other point is that, as you said, it's actually a false dichotomy, because it's more of a two-dimensional axis. You have an axis of control versus uncontrolled, and you have axis of natural versus unnatural, and you can have also situations where you have a controlled but natural behavior, so behaviors that are highly repeatable, highly reproducible, but also highly natural. For example, birdsong, or the hunting sequence of the bat, or many other, like escape of a cockroach, or many other behaviors that are done relatively stereotypically by animals, and so they're very amenable to study, but they are very natural. So I think those are the type of behaviors that are particularly important that we look at as neuroscientists.
Mm-hmm. Yeah, I'm just kind of curious on the topic of natural neuroscience, that kind of stuff. What do you want to do next? Do you have like a big new project that, advanced this to a whole new level, or I'm just curious how it sounds like you've been thinking about this quite a lot, what you want to do next and making these more natural tasks.
First of all, I think we are going in my lab to more and more naturalistic experiments. So, on the social side, groups of animals, more and more natural interactions. Also, the technological development helps a lot in that, because we are developing these neural loggers that we have been developing. They are becoming smaller and smaller, so it's easier and easier for the animals to carry them. So all of this helps a lot on the practical side with the experiments. More complex and naturalistic navigation. I think down the road it'll be very interesting to look at study the brain outdoors. That'll be sort of a dream experiment. And generally, I would say we are moving more and more into more natural experiment, while still maintaining control and reproducibility. So it's very important to stress we are not doing experiments where animals are doing random stuff. We do have them repeat some behaviors, do some control manipulations, but have the experiments in a more naturalistic situation than normally done in laboratory studies.
Mm-hmm. So at the end of each interview, I asked my guests the same kind of 3 questions. The first is, what's a book or paper you think more people should read? This can be famous, completely unknown, old, new, just something you think more people should read.
It's not a particular paper, but I think the old papers I would highly recommend. I remember to this day when I was a PhD student, I took this course about reading these old papers. So by old, I mean Hodgkin and Huxley, Hubel and Weasel, the magical number 7 plus or minus 2, what the frog's eye tells the frog's brain, all those classical papers from the 50s and 60s. First of all, they're a joy to read. You learn about them in courses, but to read the original is really interesting. They're written in completely different language and in a completely different way than things are written nowadays. And it's very interesting. Like, you read Hodgkin and Huxley, and in one of their papers, they, spend several pages, literally, asking the readers forgiveness for the fact that they did the experiment not in body temperature, but in room temperature, and what would that entail? Because of course, all the kinetics is very much temperature dependent. So, but they literally spend like 2 or 3 pages, saying, well, it's problematic and we're sorry and we have this limit. It's like, where would you see in any paper today somebody full 2 printed pages, acknowledging a problem like that? I mean, this is really amazing to read. So I would tell students, go read original classics. It's just interesting. If you won't learn anything beyond what's written in the textbook, you'll just learn about a different style of research and about how to write things. It's these people got Nobel Prizes for good reasons, and it's highly recommended to read those original papers.
Yeah, the writing style, how that changed over the years, I find it quite interesting too. I mean, I haven't read any of the specific ones you mentioned, but I remember once I just kind of can't remember why, but for some reason I was just like, I wonder what the paper is like where Turing wrote about the Turing test, right? Where he wrote about that. And so I just randomly found it and then started, I think I read the first few pages, and what really surprised me was that, this was a computer scientist or mathematician writing kind of philosophy. And I thought it was going to be super complicated and technical, but the first 2-3 pages that I read were just so easy to read. It was so clearly written. I was really amazed by it. Yeah, it makes me wonder sometimes why exactly some of that got lost.
Yeah, I totally agree.
Final question is advice for PhD students and postdocs. So kind of people on that transition. Any advice you tell them?
Yes, I think one advice is to find your own way, find your own niche. I mean, I did it when I switched to the bat, and it proved very successful. If you're looking for doing something that is different from others, it's often an advantage. It's easy to look at what everybody are doing and say, I want to do the same thing. It seems to work. Everybody are doing mice, optogenetics, whatever nowadays. So, but there are thousands of people like you, and actually doing something different could be a different species, could be a different question, a different brain area, something different stands out. And I actually think that's very important. On the other hand, it's also important not to go to something very esoteric. I mean, I love, for example, neurotology, where people look at particular interesting critters. But some of the animals that people study there are just not, most neuroscientists don't care about this. So if you're a neurologist, if you want to understand particular species, great, go for it. But if you want to stay closer to the mainstream of neuroscience, then I would say trying to find a species or a question for which many researchers would find the research interesting. For example, my studying the bats, although I study a highly unusual species, on the other hand, the brain area that I study, the hippocampus, is extremely well studied. It's the second most studied brain area of all. So there's a lot of labs interested in that.
Wait, what's the first?
Well, neocortex, I would say. I once did a search at SFN, how many abstracts have, hippocampus or hippocampal in the title or abstract. This was many years ago, but there were 3,500 posters at that SFN with hippocampus or hippocampal in the abstract. That's a lot. Only cortex had more. So it's a highly studied area, and then people are interested in that. So I would say general advice, if you can find a species or a question or a direction that is unique on one hand, but does relate to the mainstream of neuroscience, that's the best place to be in, because then automatically what you do is unique, but is relevant and interesting for a lot of people. And then, there's no problem, people are interested and want to hear what you have to say. And then there's no problem to publish, no problem to get funding, etc. So this is sort of the sweet spot that I would advise people to look for. The unique but relevant sweet spot.
Yeah, it's interesting because I was wondering as you started, I was wondering how do you to some extent, your career could be one side of a survivorship bias, right? Where, let's say, lots of other people tried something in similar studies and it just didn't work out, and we don't hear about them because it never did anything interesting. And there's this one video, I think, from The Onion, which is fantastic, where they have this, obviously satirical interview with someone who studied anteaters all his life. And they're like, oh, tell me about anteaters. And the guy's just like, no, they're just boring. There's nothing interesting. I wasted my entire career on them. And just has this existential crisis on TV. But no, it was just pointless. I hate anteaters. I don't know why I did it, right? And, but so to avoid that, you think it's kind of connecting it to a mainstream thing that does address kind of fundamental questions.
I think, especially if you're interested in general questions about the brain, in general neuroscience, that's the way to go. If you're in more etologist or neurotologist at heart, and you want to understand a particular critter for its own right, great, go for it. It's just that you need to be aware that if you are studying a very esoteric species or a very esoteric question that does not relate to most of neuroscience, then you're going to remain in this niche and not break through to the mainstream. So it's sort of, it's an approach, it's an attitude and approach. If you want to do that and study anteaters or whatever, great. I love that. As I said, I grew up as a kid watching nature documentaries. When I hear talks about an esoteric species I've never heard before, it always amazes me. I love that stuff. But it really puts you away from mainstream neuroscience. If you want to remain closer to mainstream neuroscience and ask questions that are relevant for the broader community, then you need to make this connection. So this is sort of, there's a tension here, but it's doable. My research shows that it's possible. There are other people who are doing that. So, you can find these niches that are unique, but on the other hand, also relevant for the general neuroscience community. And I think that's a great place to be in, because then you're automatically you're interesting for people. What you're doing is interesting for people.
Yeah, definitely. Okay, well, with that, thank you very much.
Thank you so much. Thank you.